Sliding filament theory. The first step of sliding filament theory is the cross-bridge cycle. In order for a cross-bridge cycle to begin happening, the myosin head has to be activated by ATP, which hydrolyzed by ADP, an inorganic phosphate. The energy from the ATP activates the myosin, which allows it to move into a more cocked position. In a cross-bridge cycle, calcium will attach itself to troponin, which will cause it to change shape. When the troponin changes shape, it will allow the tropomyosin to move away from the binding sites that are on actin, which then allows the myosin head to attach to the binding sites. When the myosin head makes contact with the binding sites on actin, the myosin will release inorganic phosphate, which creates a stronger bond between the myosin and actin. This is called cross-bridge formation. Step 2. Power stroke. ADP is now released. In reaction to the ADP being released, the myosin head will now pivot and bend, pulling on the actin, sliding the thin myofilament to the middle of the sarcomere or M line. Sliding of the thin filaments continue as long as the calcium signal and adequate ATP are present. When the nerve impulses are delivered rapidly, calcium levels increase greatly, resulting in lack of relaxation of the muscle. Also, just a note, when the thin myofilaments are pulled closer to each other, this is when the whole muscle begins to be contracted. Step 3. Detachment. Once the power stroke or strokes have been completed and the nervous stimuli ceases, new ATP attaches to the myosin, causing it to detach from the actin. Once the myosin has been detached, the troponin again changes shape, and actin's myosin binding sites are again covered by tropomyosin. The result of this detachment is relaxation of the muscle, which is interesting since this step is the one that requires energy or ATP. Step 4. Cocking of myosin head. As the ATP is used for detaching the myosin from the actin, the ATP turns into ADP and phosphate, causing the myosin head to return to its pre-stroke high energy or cocked position. It's ready to take another step and attach to the actin site farther along the thin filament, creating the walking effect of the contraction. This cycle is continuously happening in our body, allowing us to function and carry out our everyday necessities.